everyone. Welcome to the final video for, let me get my pen all set up, for chapter 20. And not only is it your final chapter 20 video, it is your final video of the semester. So congratulations on making it all the way through your first semester of biology. In our last two chapter 20 videos, we covered two biotechnology processes that have been really important to the field of molecular biology. And the first process we covered was, of course, gene cloning using recombinant DNA technology. And then, I'm just going to put recomb DNA. And then the next thing that we covered in our last video was the process of the polymerase chain reaction, which we're going to abbreviate PCR, as well as the technique that we use to visualize our PCR results, which is gel electrophoresis. This last piece of technology that I want to go over, this last process, is called CRISPR gene editing. And more appropriately, you might hear it called CRISPR gene editing or just CRISPR a lot. But if we were being really complete, we would call it CRISPR-Cas gene editing. And this one is, I would say, fairly new to the field. And it is proving to be as transformative as these first two were, okay? So today, we're going to talk about gene editing using this feature of genomes called CRISPR and associated proteins, which are called Cas proteins, okay? So what is CRISPR? You may have heard of it. It's been a hot topic in the news lately. And what we discovered, the way that we found out about CRISPR was through studying archaeal and bacterial genomes. And people started noticing a feature within these genomes back in the mid-90s to early 2000s. What they found was that there would be a region. So remember, if we're talking about a bacterium, again, as I've told you a million times, they have single circular chromosomes. And there would be a region on this chromosome where you would find a promoter. So I'm just going to put promoter here, P. And then you would find these repeats, which I'm going to abbreviate R, with a spacer set of sequence in between. And then there'd be another repeat and another spacer sequence. And I'm putting that in quotes because I'm going to tell you what that spacer sequence turned out to be. And this would go on for quite some time, with the repeats being identical to one another and the spacer sequences being unique. And so for a while, we weren't really sure what this arrangement meant. Why was it there? But what we found is that this entire set of DNA could be transcribed as a single ginormous RNA that was called a cRNA. And that CR stands for CRISPR. Now, what does CRISPR stand for? Well, CRISPR stands for this. It stands for clustered regularly interspersed hope I'm spelling all this right. You know I'm a terrible speller. Interspersed short palindromic, meaning they're the same forwards and backwards, palindromic repeats. Truly one of the most cumbersome acronyms of all time. So you can see that this, if you put this together, this is in fact the acronym CRISPR. And while it is like super cumbersome and maybe not the greatest, you can see that it does accurately describe what researchers were seeing. So these are repeats that are clustered, okay? They're short, it turns out. They are fairly regular. If you examine the sequence, they are the same forward and backwards. So this term CRISPR really describes a locus on bacterial genomes. That's really what CRISPR, or our archaeal genomes, okay, what it describes. And so like I said, you could transcribe this. You would have your RNA polymerase bind at the promoter, and then it would copy this into one ginormous long CRISPR RNA. Here we have a cRNA. So here you can see we have our CRISPR locus with a promoter, and then these are our repeats, and these are our spacers in different colors. Here we've transcribed this DNA into RNA right here, okay? So here's the 5' area, and then we have our repeats and our spacers. And then what happens is this one large RNA gets chopped up so that we have little individual RNAs that have part of the repeat on either side with the spacer in the middle. Part of the repeat on either side with the spacer in the middle. And here again. So what was going on with these repeats with spacers in the middle? Well, here is what was happening. So let's just draw ourselves a bacterium, okay? Now, y'all know, and don't forget to draw yourself a single circular chromosome on your bacterium, okay? Now, y'all know from previous lectures that these bacteria can be infected by these spaceship-looking viruses called bacteriophages, right? And what bacteriophages do is they like to inject their genetic material, as all viruses do, into cells, right? That's how they infect cells. Well, what bacteria can do is they can chop up this viral DNA. We've actually talked about this before. They can use things like restriction enzymes to chop this up. And what these bacteria were doing is they were taking a little chunk of this chopped up viral DNA and they were storing it in their chromosome, okay? And if a new virus came along and infected the cell, it would chop up the DNA and it would store just a little bit. And then if a third virus came along and the bacteria would chop up its DNA and it would store just a little bit. Hopefully you're getting the idea that what was happening is, we go back here, these spacers that we see here, or these spacers that we see here, what these spacers consist of are little bits of viral DNA that the bacteria was storing. The reason that this bacteria is storing this DNA is to help it recognize that DNA later. So here's what happens. You go through and you transcribe this CRISPR RNA and you break it down. So let's just draw ourselves. I'll draw myself some little RNAs here. We'll make it, this one be the orange one. And we'll have this one be the purple one. And we'll have this one be the green one. Okay. All right. So what we've done is we've transcribed this into one long RNA and then we chopped it up so that we have a repeat on either side with some of that spacer DNA in the middle. Now what's going to happen is that each of these individual little CRISPR RNAs are going to associate with a protein that I'm just going to draw a circle around it here. These proteins are called Cas proteins, and Cas just stands for CRISPR associated proteins, okay? So CRISPR associated proteins. That's where we get that name. So these Cas proteins, what they do with their little guide RNAs, we're going to call this a guide RNA. They roam about the cell, patrolling the cell, seeing if they can match up this viral RNA guide that they have with any viral genetic material. So let's just say we have another infection. Okay, so we're going to have a little bacteriophage. It's going to come along and we'll make it be the green one. It's going to come along and it's going to infect. Well, this Cas protein is going to come along and it's going to say, wait a minute, this gene here matches this gene here. If this Cas protein finds a match, it cuts up this viral DNA and thereby it inactivates the viral DNA, right? By cutting it up and activates. So essentially what's happening here is this CRISPR-Cas system is acting like a bacterial immune system. So bacterial immune system. I'm going to put system, and I'm going to abbreviate that. Okay, so here is a close-up of this CRISPR-Cas situation. Here is in green, this is the Cas protein. Here is our, right here, actually, sorry, this right here in the blue, that is our guide mRNA, okay? So that's our guide RNA. Don't worry about this. This is a level of detail that I don't expect you to know. But what I want you to know is that this guide RNA came from this region here, where the bacteria had been storing little bits of viral genetic information. So the Cas protein comes along and it's looking for DNA that matches this RNA. And the way that it does that is it actually separates the two DNA strands and it tries to see whether this guide RNA here matches either of those strands. And if it does, you can see we've got some little scissors here. It's going to cut this guy up to inactivate that viral DNA. Here's a diagram that sort of summarizes what I just told you. So first you have a bacteria that is infected by this virus. This viral DNA comes in, gets chopped up, and then it gets stored in the bacterial chromosome as a new spacer sequence, okay? So that's what this says here. A new spacer is derived from the virus and it's integrated into the CRISPR sequence. So here in black, those are your repeats. And then the little color blocks are your spacers. And again, each one of these is derived from some viral DNA when the cell became infected. Now we're going to transcribe this and we're going to cut that up into little pieces of CRISPR RNA and we're going to associate it with a Cas protein. If this CRISPR RNA then matches viral DNA from a later infection, we're going to chop it up, okay? So here's the thing, the initial native function of this CRISPR-Cas system was bacterial immunity. But we have figured out that we can use this CRISPR-Cas system for gene editing. What we can do in the laboratory is we can take little sections of repeat sequence, and then what we can do is, let's see, I'll just pick a new color. I'll use this aqua color. We can insert a sequence for a gene that we are interested in either inactivating or changing. So this is a gene sequence that we insert, and we'll call this an insert for a gene of interest. So GOI is gene of interest. So then what we can do is we can allow this This is what we're going to call our guide RNA, okay? We can allow this to then associate with a Cas protein. And we can send it into the nucleus of a cell that we're working with. You might be working with plant cells and maybe you want to genetically engineer some plants. You might be working with some animal cells and you want to genetically engineer those animals. You might even be working with human cells. So you can send this Cas CRISPR situation into those cells, and if this lines up with a gene of interest, this Cas protein will cut the DNA. Okay, so let's look at it here. So what we have is some DNA. And this DNA is, let's just say that it is in the cell of a mushroom. We're going to genetically modify a mushroom. And the gene that we're interested in destroying is the gene that causes these mushrooms to turn brown. I don't know if you've ever purchased fresh mushrooms, but they start to go brown in your refrigerator pretty easily. So this is just double-stranded DNA. And this gene is the gene that causes mushroom browning. Browning, sorry. Okay, so what we're going to do is we're going to send in to this mushroom a Cas protein with a guide RNA, and it's going to search in the DNA of this mushroom and it's going to look for this gene. If this RNA here matches this DNA here, and again, the way the Cas protein will figure that out is it'll push apart these two strands and try to line up this RNA. If it lines up, what it'll do is it will cut the DNA as you see represented here. Now, in your cells are not going to tolerate some cut DNA. They're going to try to repair this cut. Here's where the inactivation comes in, because in repairing this cut, what happens is, remember, we've got these two strands of DNA, let me draw them like this, that we've just cut. Now, normally the way that your cell would want to repair this double-stranded break in an ideal situation, there would be, like, on one of these strands, there would still be some information, and then your enzymes would use this information as a template to fill in this gap, or vice versa. But we have made a double-stranded break. There's no information to fill in this gap. So the repair machinery starts filling in this gap with whatever sequence it can kind of make up to fill in this area. So we're just going to put, you know, this as a made up sequence. And because this sequence is imperfect and made up, it's going to create a mutation in the DNA, okay? So that is a problem. This mutation is a problem. It can cause insertions, it can cause deletions, it can cause frame shifting. Whatever exon this occurs in is going to end up being damaged. And that damage can result in inactivation of the gene. This process where we are trying to repair, this process where we are trying to repair a double-stranded break like that is called non-homologous end joining, or NHEG. And again, the problem with non-homologous end joining is that we don't have any information to help us fill in this gap. So your enzymes just do the best they can in filling in this gap. This is a very mistake-prone process, okay? So in this way, we have edited this gene using CRISPR And what we have done is we have destroyed or inactivated this gene. We use this technique a lot called gene knockout, inactivated. We use this technique a lot called gene knockout in the laboratory. So we might take a fish or a mouse or whatever organism we're looking at. And sometimes the way to figure out what genes do is to inactivate them and then see how that impacts the physiology of the organism you're working with. So using CRISPR to edit genes to knock things out is a really important research tool. It can also be a really important tool in genetically modifying organisms. So I told you already that we've used this tool to knock out, and that's usually the terminology that we use. We've used this tool to knock out the gene that causes browning in mushrooms. We've also used this knockout tool to make what are called PRRSV resistant pigs, okay? So, you know, pigs are an important livestock that we use to feed people. And so obviously we have an interest in maintaining pig health. So pigs are susceptible to this virus that's called porcine respiratory and reproductive, and I'm going to abbreviate that, repro syndrome virus. Now, I'll be honest, this isn't a virus that I know a lot about, but I do know that as a virus, what it does is what all viruses do. It tries to find a place on the cell where it can bind. And so since this is a eukaryotic cell, I know that it's going to have a receptor. And so this virus particle is going to want to dock, and I'm drawing it kind of like a coronavirus. I'm sure it has proteins on it that are receptor proteins that are going to allow it to dock with the receptor on the surface of this pig's cell. Well, what we've done is we have used CRISPR/Cas editing to knock out this receptor. And if I'm not mistaken, and I need to go back and verify this, but I'm pretty sure that this knockout is only in the region that binds to the virus, and that the rest of this receptor remains intact, because they only used the CRISPR-Cas system to inactivate the exon that coded for the region where the virus binds. So what that did is it allowed us to make pigs that were resistant to this virus, but otherwise were physiologically okie dokie. They lived just fine, okay? So we can use this CRISPR-Cas technology to knock out genes. We can also use this CRISPR-Cas technology to alter genes, okay? And in this example, I want to talk about a disease called sickle cell anemia. Anemia, yeah, I guess I spelled that right. Okay, so what happens in sickle cell anemia is normally your red blood cells, which are shaped kind of like donuts, they have like a little, sort of like a little dip in the middle, because they are actually sort of more disc shaped than round. If you were to cross section them, they look kind of like this. So that's the cross section. That's the normal shape of your red blood cells. Sickled red blood cells start to take on irregular shapes that are often sort of like this, or I'm going to show you a picture here. This is a sickled red blood cell. You can see that it's not the right shape. That is a sickled red blood cell, that one as well. You can see that those are just the wrong shape, as opposed to in this picture, I don't know if you can make out, like all of these really nice red blood cells, like this one is normal, and that one is normal, and that one is normal. So these sickled red blood cells have an abnormal shape, and there's two problems with this. First of all, they don't fit well. They do not fit well in your capillaries. They can get all hung up and stuck. The other big thing, the reason that they have this odd shape is they do not bind oxygen well. And that is because, there is a mutation in the gene for hemoglobin. It's actually the beta hemoglobin gene. That's a G, beta hemoglobin gene. So the deal is, we actually know exactly which gene is the problem. So what we can do is we can use our CRISPR-Cas editing system. And in this case, what we're going to do is we're going to make not one, but 2 cuts, okay? So let me draw this a little better in a way that's easier to understand. We're going to take our DNA, and this is our gene for the beta hemoglobin here, okay? So I'm going to draw it like this. 2 strands, okay? I'm drawing 2 strands here because I want to show that we're going to cut both strands. And what we're going to do is we're going to use our Cas system to cut around the defective region. So we're going to cut here, and we're going to cut here. And when we do that, these little cuts are going to float. These little pieces are going to float away. So we have a big open region that we're going to need to fill up, okay? So we need to fill that region up. Now, if we just let non-homologous end joining, like I showed you in our last slide, if we let that fill up this region, we're going to be in a pickle. That is going to fill it up with erroneous sequence information. But what we can instead do is we can provide the cell with something to look at to fill in this region. We can actually put in a little bit of DNA with the correct sequence. So this would be the proper sequence for the sickle cell anemia gene. So I'm just going to draw down here. I'm going to draw some double-stranded DNA that spans this area, and I'm running out of space. Let me make this smaller or something. Let me redraw this and make it smaller so we can have plenty of space. So up here, we'll have our cut double-stranded DNA. And what we're interested in doing is in filling, let me make this back here. And we're interested in filling in that gap. So what we're going to do is we are actually going to put into the cell a little bit of DNA that would correspond to this region that needs to be filled up. And we're also going to have a little bit of overlap here that corresponds to these regions, okay? So what this is going to be treated as, the cell is going to look at this and say, this kind of is almost like a homologous chromosome, and we can use this homologous chromosome information to fill in this region, okay? This is called homolog directed repair. This is actually like when your cells are crossing over during meiosis, and sometimes there will be a double-stranded break. That's exactly what your cells do. They look at the homologous chromosome to see if there's any information it can use to copy, and if there is, then it will fill that gap in with the information it's using to copy. So it's almost like looking at someone else's paper during an exam, right? So I'm going to pause this, and we're going to look at a video that describes this process in a way that might be a little more clear. So.
Every cell in our body contains a copy of our genome, over 20,000 genes, 3 billion letters of DNA. DNA consists of 2 strands twisted into a double helix and held together by a simple pairing rule. A pairs with T, and G pairs with C. Our genes shape who we are as individuals and as a species. Genes also have profound effects on health, and thanks to advances in DNA sequencing, researchers have identified thousands of genes that affect our risk of disease. To understand how genes work, researchers need ways to control them. Changing genes in living cells is not easy, but recently a new method has been developed that promises to dramatically improve our ability to edit the DNA of any species, including humans. The CRISPR method is based on a natural system used by bacteria to protect themselves from infection by viruses. When the bacterium detects the presence of virus DNA, it produces two types of short RNA, one of which contains a sequence that matches that of the invading virus. These two RNAs form a complex with a protein called Cas9. Cas9 is a nuclease, a type of enzyme that can cut DNA. When the matching sequence, known as a guide RNA, finds its target within the viral genome, the Cas9 cuts the target DNA, disabling the virus. Over the past few years, researchers studying this system realized that it could be engineered to cut not just viral DNA, but any DNA sequence at a precisely chosen location by changing the guide RNA to match the target. And this can be done not just in a test tube, but also within the nucleus of a living cell. Once inside the nucleus, the resulting complex will lock onto a short sequence known as the PAM. The Cas9 will unzip the DNA and match it to its target RNA. If the match is complete, the Cas9 will use two tiny molecular scissors to cut the DNA. When this happens, the cell tries to repair the cut, but the repair process is error-prone, leading to mutations that can disable the gene, allowing researchers to understand its function. These mutations are random, but sometimes researchers need to be more precise, for example, by replacing a mutant gene with a healthy copy. This can be done by adding another piece of DNA that carries the desired sequence. Once the CRISPR system has made a cut, this DNA template can pair up with the cut ends, recombining and replacing the original sequence with the new version. All this can be done in cultured cells, including stem cells that can give rise to many different cell types. It can also be done in a fertilized egg, allowing the creation of transgenic animals with targeted mutations. And unlike previous methods, CRISPR can be used to target many genes at once, a big advantage for studying complex human diseases that are caused not by a single mutation, but by many genes acting together. These methods are being improved rapidly and will have many applications in basic research, in drug development, in agriculture, and perhaps eventually for treating human patients with genetic disease.
Okay, so this, as the video pointed out, this is a technique that is still very much in development. We use it in research a lot, but it's not been put into widespread use in human medicine. However, one particular disease that is showing great promise with this CRISPR-Cas editing is sickle cell disease. So there is a patient who, if you look up New York Times articles, she's been actively treated using CRISPR-Cas9 editing. And this is an example of what her blood looked like before and after the editing. So each of these little red arrows here, are pointing to a cell that has an abnormal shape. The word we would often use is morphology, M-O-R. So these are cells that have abnormal morphologies. Morphology is just a term that says study of shape, right? So morph is shape, ology is study. So these are abnormal morphologies on each of these cells. This was before this woman's genes were edited using CRISPR. After they edited her genes, check it out. You don't see nearly as many of those red arrows, do you? I'm going to erase my marks that I was showing you earlier so you can see that these are far more, there's a whole lot more normal cells here than there were here. So what they essentially did, let me see if I left myself, let me make myself some, okay, yeah, let me insert here. I want to insert a blank slide so I can kind of just write out for you what they did. Essentially, hang on, oop, there we go. What they did is they went into this woman's cells, or her, actually they went into her bone marrow, okay? So this was step one. They got out some of her bone marrow. Now, if you're not sure why that's relevant, you may be aware that bone marrow inside the interior of your bones, that is actually the tissue that makes blood cells. So you might have heard of someone having a bone marrow transplant. That would be like if you had someone that had leukemia, which is a cancer of your blood cells. If you had someone that had a leukemia, then what they would do is they would try to find a donor whose their tissue was a match. And so they would take bone marrow from the donor and then insert it into the recipient to try and get healthy blood cells made. So what they did in this particular individual with sickle cell anemia was they went into her bones and withdrew some of her bone marrow. Then they edited these bone marrow cells using the CRISPR-Cas system, okay? And you may have heard in the video, the Cas that we use is Cas9. There are actually a number of CRISPR-associated proteins. Cas9 is the one that we use most frequently. So they edited her cells using the CRISPR-Cas9 system to insert the correct sequence, the correct hemoglobin sequence. And I'm just going to put heme sequence, okay? And I'm going to put correct there. All right, then they put back her edited cells, and then they waited to see if these edited cells would make normal blood cells. And they have had tremendously positive results, okay? Now, the important thing to note here is that you have to know exactly what gene is causing your disease, and you have to know precisely what and where the mutation is that is causing the disease in order for this to work. So this is actually sort of a technology, like I said, that's under development. And a lot of it has to do with the fact that we still don't have great information on all the mutations that cause the genetic diseases that we know of in humans, okay? Now, one question you might be asking yourself is, how do you get this CRISPR-Cas system into cells? Well, it turns out that you already know the answer. And the answer lies in our plasmids. And if you remember, plasmids are little circular pieces of DNA that we get from bacteria, okay? I think I might have said in the last video, I might have misspoke at the beginning and said that plasmids come from viruses. They do not, they come from bacteria, okay? So remember that you have these little circular pieces of DNA that you get from bacteria, and that you can use restriction enzymes to cut these guys, right? So you can have these little cut plasmids. Well, what we've been able to do is to design plasmids where we insert the CRISPR RNA that we're interested in, and then we also, and here's the nifty bit, we also insert the gene for the Cas protein. So here's our Cas DNA. Here is the CRISPR guide RNA sequence. Now we can take this plasmid and we can insert it into cells, okay? And remember from our first video from chapter 20, the way that you can insert plasmids into cells is you just take those cells and you make their membranes porous. Remember how we did that with the bacteria? So I'm gonna draw a nucleus here because we're dealing with like human cells in this case. But once you make those membranes porous using chemicals or using electricity, then this plasmid can slip right in. And then what you can do is use the machinery of your cells to then transcribe and translate, I should say, the genes on this plasmid. And so when you do that, you're tricking your cell into making its own Cas proteins, and you're tricking your cell into making its own CRISPR RNA. And so that you can get the whole system into your cells. You can do this, like I said, by making the membranes porous. You can actually also engineer a virus to do this as well, to inject this plasmid into cells. But once you get this plasmid into your cells, then the cells will make the Cas and the CRISPR all on their own, okay? So here is an example of how you could use a virus to get this plasmid into your cells. Turns out that there are some viruses that are benign. In other words, they don't cause disease. The word benign means not harmful. So if you have a benign tumor, you have a non-harmful tumor. A benign virus is a non-harmful virus. And so in this example, you see a virus that's attaching to the surface of a cell. It's getting into a cell, and it's leaving behind, here's its genetic material. So in this case, it's our plasmid. It's leaving behind our plasmid, and that plasmid then will be translated and transcribed inside the cell to make our very own Cas and CRISPR products. Now, the interesting thing is this idea of inserting the Cas gene and the CRISPR gene have a third application, and that third application is you could use it to make something called a gene drive, okay? Let me show you a quick video on how these gene drives work as well.
Historically, scientists have been able to alter the traits of domesticated plants and animals, but have not been able to do this to wild populations. Here we use mosquitoes as an example to explain why most genome alterations designed by humans don't persist in nature, and how a recently proposed technology could change that. The transgenic mosquito, tinted blue, has an altered gene inserted into one of its chromosomes. When it mates with the wild-type mosquito, each parent contributes one copy of each chromosome to their offspring. Thus, only 50% of offspring will carry the altered gene, while the other half will inherit the wild-type version from both parents. Even if the altered gene doesn't reduce the likelihood of each mosquito surviving and reproducing, it may persist at a low frequency in the ocean of wild mosquitoes, or it might go extinct after several generations of especially unlucky inheritance. This process is what keeps us from altering wild mosquitoes to prevent them from transmitting diseases such as malaria or dengue. A team led by Kevin Esvelt at the Wyss Institute, the Harvard Medical School, and the Harvard School of Public Health has now outlined a way to build gene drives that can improve the odds that almost any altered gene will be inherited, potentially allowing them to spread through even wild populations. The proposal relies on the CRISPR-Cas9 system, a new genome editing technology co-developed by the same researchers at the Wyss. Gene drive mosquitoes carry both the altered gene, the genes for the Cas9 enzyme, and several guide RNAs that tell it where to cut. When passed to offspring, the guide RNAs direct Cas9 to cut the wild-type version of the gene inherited from the wild-type parent. The cell then copies both the altered gene and the drive when it repairs the damage. And because the mosquito now has two identical copies, one on each chromosome, all of its offspring will inherit the alteration in the gene drive. This same process will be repeated in subsequent generations, causing the alteration in the gene drive to spread through the population.
So in the video, they were talking about a gene drive for this guy here. This is the Anopheles mosquito. And the reason that this guy is important is because this is the carrier of malaria. And so for many, many years, we've been trying to devise ways to control the population of these mosquitoes, because if we can control their population, we can reduce the incidence of malaria. So these mosquitoes represent a public health threat. Malaria is a very, very damaging disease, okay? Now, you guys know that mosquitoes are pests and they reproduce very fast and they're really hard to get rid of. But if you can engineer a mosquito so that it has in its DNA the gene for CRISPR, the gene for the Cas cutting enzyme, and then what these folks engineered was a gene alteration that caused sterility in female mosquitoes, okay? So this became inserted into some mosquitoes, and then as the cells translated and transcribed this, the first thing it would do is, if it was only on one chromosome, it would alter the second chromosome as well. That means that all of these mosquitoes became homozygous. Now, when these mosquitoes go to reproduce, they're going to give one copy of each chromosome to their gametes, and that means that all of their gametes are going to have this altered gene in them. This altered gene then can get into their offspring, where it will edit their genes and make them a homozygote, and then it'll get into their offspring where it will make them homozygotes. In other words, this editing guarantees, did I spell that right? I don't think I did. Guarantees that the sterility gene gets passed on. And so some of these mosquitoes are going to die out, and the ones that remain that are still in the wild population, when they mate with these altered mosquitoes, they're going to die out as well. And so over time, the entire population would die out. We have actually accomplished this in the laboratory. Now, you may be asking yourself, if that's the case, and we've done this in the laboratory, why haven't we deployed this? Well, there are some ethical concerns, okay? So some of the concerns are that we're not exactly sure what the environmental consequence of eliminating mosquitoes, consequence of eliminating these mosquitoes would be, okay? So we're not sure about that. We do worry a little bit about gene transfer. So what if one of these mosquitoes that was genetically engineered was able to hybridize with a closely related insect and pass on the sterility gene? Well, now you've potentially knocked out not just mosquitoes, but another organism as well. Another concern in deploying this technology is that in terms of the ease of doing this, this is kind of easy to do. And so there's a fear that if we start immediately deploying this CRISPR technology, then you could have a situation where anybody with minimal knowledge tries to do these edits. And there's concern that it could lead to mistaken edits. So it's kind of almost a bioterrorism issue or concern as well. But the good news is, despite these ethical concerns, things that should make you feel safe about CRISPR editing are, one, that it turns out in order for you to be able to pass this gene on effectively throughout a population, you have to have a very short generation time. So this is only going to work in things like insects that multiply and reproduce every couple of days. You couldn't eradicate the human species very rapidly because we don't multiply that quickly. You couldn't eradicate elephants very rapidly because they don't multiply that quickly. The second thing that should give you a little bit of, alleviate your anxiety a little bit about any of this stuff, is that any mistakes that get made should, in theory, be reversible. So in the same way that we inserted this sterility gene, if we wanted to reverse these effects, we would just like in another set of mosquitoes, insert the correct gene to try and rescue this effect. And then the last thing is, it's really hard to associate specific genes with specific functions. So while I said that in theory, it's pretty easy to do in the laboratory, that assumes that you know something about the genes that you're working with. And the truth is, more often than not, we don't know much about the genes that we're working with. So this is sort of the direction that molecular biology is taking as it moves forward using some of the concepts that we've already learned this semester in this class. This brings us to the end of the new material that you will have in Biology 105. From this point on, regardless of which semester you're watching this, you should be studying using some of the study guide concepts that I've given you. I should have given you a study guide at this point that outlines the things that you're going to be responsible for on your final exam. Have a look at all of that. I hope you learned a lot this semester, and I hope to talk to you soon. Bye-bye.